gente, hoje o nosso grupo vai entrevistar o Dr. Brune Levine, da Universidade da Pensilvânia, que desenvolveu grandes avanços na terapia gênica contra o câncer. E a gente vai entender um pouco das suas motivações, de como foi esse processo no desenvolvimento dessa terapia.
Então nós vamos entender principalmente como esses genes têm acesso à célula e a maneira que o Dr. Brune programou os receptores do tipo CAR-T para que eles tivessem acesso às células cancerosas e dessa forma compreender o desenvolvimento total da sua pesquisa.
Hi, Mr. Levine. Good morning.
Good morning. How are you?
I'm fine. My name is Letícia.
Bom dia.
Bom dia. And today we are going to ask you some questions about your job. But at first, we want to know some things about your choices. Because nowadays, new treatments are being widely discussed, and one of them is gene therapy. And we want to know what motivated you to take it, to dedicate yourself to take up research on gene and cell therapy.
Well, I, as an undergrad, was majoring in biology, but asking my professors, what do you see as the up-and-coming field for the next 20 or 30 years? And a couple of them said immunology. So when I went to grad school, I went to a program in immunology and began working on T cells for my thesis work, and as background was reading papers by a guy named Carl June. So I applied to his laboratory for a postdoctoral fellowship. I'd been interested in cell culture and immunology and wasn't really aware because there wasn't that field of cell and gene therapy yet. But I went to Carl June's lab, and one of my postdoctoral projects, have any of you used the Dynel anti-CD3, anti-CD28 beads to grow T cells? Well, it's a way to grow T cells. That was one of my postdoctoral projects, and we found that very efficiently expanded T cells in the lab. And so then Carl June asked me how would I like to start a lab that grew T cells ex vivo for a clinical trial of adoptive immunotherapy. And that's really just what I wanted to do. So that began in the late 1990s, and we conducted a clinical trial. We moved to the University of Pennsylvania in 1999 and began many other clinical trials, not just in HIV, but in cancer. So I don't know if that directly answered your question, Leticia, but happy to follow up.
Mr. Levine, while we were reading about your job, the treatment with CAR T cells impressed us. And we understood that some T lymphocyte cells are removed from the patients, and after that they are genetically modified and reprogrammed in lab. And they recognize the tumors because of the antigen CD19, and they target tumors. And after that, after this modification, they are reintroduced into the patient's body. But could you explain us a little bit more about it and tell us which challenges you and your team faced along the development of CAR T therapy related to cancer treatments?
Little additional piece in background about what we were doing. So normally in the body, T cells receive their signals from dendritic cells that present antigens. What the beads are is a substitute for dendritic cells that's off-the-shelf way to stimulate T cells. By having two antibodies, anti-CD3 binds to a component of the T cell receptor to deliver signal 1, and anti-CD28 binds to the main co-stimulatory receptor. So that was one technical hurdle, being able to grow T cells in the lab. A second was the design of the chimeric antigen receptor. So this is basically a way to redirect T cells to find tumor or leukemia cells or any antigen and it's an antibody on the outside and T cell signaling domains on the inside. And there are a number of considerations for design of the chimeric antigen receptor, or CAR. Then you need a way to get that gene inside the cell. Most often we're using a viral vector. In the very beginning, people were using a retrovirus that was derived from a murine leukemia virus, but now most groups are using a lentivirus, which is derived from HIV. It's disabled, it cannot cause disease. Its only role is to deliver RNA that is reverse transcribed into DNA and permanently integrated into the genome. So those are the main three technical pieces, and you work out this technology in the research lab at research scale with research reagents. And how do you translate that up from millions of cells to billions of cells, from research reagents to clinical reagents? And then the next hurdle is to submit to the regulatory authority. In Brazil, I think it's Anvisa, right? So you have a dossier that you prepare to submit to the FDA or to Anvisa to conduct the clinical trial. You need the clinician expertise, the laboratory to produce the cells, quality control laboratory to test the cells, and then also after the patients at baseline and after patients receive the CAR T cells, taking samples from them to come back to a laboratory to do the correlative studies. So when patients respond, you want to better understand mechanism of action. When they don't respond, to understand why that was, and so you can go back to the laboratory and improve the manufacturing, the design of the CAR to enhance potency or something else.
So thank you so much. And now Ana Clara will ask you some other questions about your job.
Hello, Mr. Levine. We're so glad to be with you today. And I have some questions for you. And one of them are, which characteristics of this type of cancer, like leukemia, have you seen that possibilities the treatment with CAR T cells? And what's differentiated this kind of cancer from other ones that is not possible to use this type of treatment?
Okay, so we have in the US and in Europe approval of 6 different CAR T cell therapies directed against 2 targets. One is CD19 that is present on leukemias and lymphomas, and another target is BCMA, B cell maturation antigen, that is present on plasma cells and myeloma. So the initial approvals have occurred in relapsed refractory disease, but there have been 2 clinical trials for 2 products for approval in second-line lymphoma, so earlier in disease. There have also been approvals in different types of lymphoma, follicular lymphoma, and mantle cell lymphoma. So those are all blood cancers, and the challenge has been in solid cancers. One part of that challenge is finding the right target, because CD19 and BCMA are on B cells and plasma cells, but they're really not on any other tissues. In solid cancers, most of the targets may be bright on tumor cells, but expressed at lower levels on other tissues. So then you have to worry about on-target but off-tumor toxicity. There are ways with dealing that with affinity of the antibody or potentially switches. So that's one challenge. The second challenge is the solid tumor microenvironment is generally much more immunosuppressive, which means that you have to think of tumors from an evolutionary perspective. Those that have proliferated, have survived, have generated tumors, have evolved ways to hide from or suppress the immune system. And there's ways to get around that that we're learning about. And then a third challenge, maybe not discussed as much, is geometry. In blood cancers, it's generally a single cell suspension. Some lymphomas, there are nodules, but it's generally accessible in blood and bone marrow. In solid tumors, you have a big mass or masses, and how do the T cells or NK cells or other immune cells not only traffic to that tumor, but get inside to kill the tumor cells? Recently, there have been some encouraging reports in glioblastoma or brain cancer from 3 separate centers. At the American Society for Clinical Oncology meeting over the past several days, there is one report that is getting a lot of attention in liver cancer, targeting an antigen called GPC3. So we're seeing some progress in solid tumors. People over the past several years have been asking, well, you have these approvals in the blood cancers. When are we going to see progress in solid tumors? And my answer is, we're seeing some progress, but it's slower than we would like, but there are reasons for that. I hope that answered your question.
Oh, yes, thank you very much. We continue to talking about the treatment with CAR cells. In case of metastasis, is it possible to make the cancer go into the remission with this treatment?
Yes, so we have long-term durable remissions. The complete response rate does vary by the disease. For example, in pediatric acute lymphoid leukemia, initially, there's a 90% complete response rate. At 2 years, that's about 65, 67%. In lymphoma, it's a little bit less. In myeloma, it's the same as lymphoma, maybe a little bit less, but not as durable as the responses in lymphoma. And there have also been clinical trials in chronic lymphoid leukemia. We have treated patients, and that is more challenging still. So that's why I say having a laboratory and infrastructure that takes samples from the patients to look at who is responding and who is not responding and why is important, so that we can increase the response rates.
Thank you so much, Mr. Levine.
Okay, thank you. Good to speak with you.